Big statement up there right now, the most powerful small molecule drug discovery platform. Well, we've come up with a new technology, and we think we can be the Illumina for industrial scale microbiome mining. And when you have direct access to the small molecules in nature on the kind of scale this technology allows, you can go get what are the mysteries of cellular communication. And that's what we're going to talk about today. So a little bit about me. I started my first company in 1988. It went on to hit the Inc. 500 5 years in a row, generated more than $435 million. Ended up getting a lot of awards along the process, ended up using it more or less for my platform to launch other technologies and companies. The technologies that we've launched included ARPA-E awards, DARPA, US Air Force, NIH. The company Biosortia is the technology company of industrial scale microbiome mining. We're still focused on what people in life sciences need, especially pharmaceutical discovery. So our chief scientist comes straight from that industry, worked with Pfizer and a number of other companies, and that's Dr. Guy Carter. Our board is heavy on the pharmaceutical industry with 4 CEOs within the pharmaceutical industry. Let's talk about the real problem. The real problem, there is no direct access to the best starting points in drug discovery. Now that sounds a little bit odd, but unculturable microbes are the best sources, but less than 1% of the microbes can be grown in a lab. Yet from that 1%, 50% of all drugs on the shelf came from that. Now, the result has essentially been when you don't have direct access to these kind of molecules, you end up creating enormous cost in discovery. And then those costs, when you start with the wrong material, fall out of the system, which means it creates cost in preclinical and the clinical aspects as well. That's a problem. So what did we do in order to have that direct access that nobody else has ever been able to do? Well, there's a couple issues here. One, people never knew how important it was to get at microbiomes of the world. Well, microbiomes of the world hold the cellular signaling of microbes. Reality of it is everyone did culturing over the last 70 years. When they did try to get directly to it, they were looking at water and wastewater plants and sludge. It takes a very specific technology that took us 10 years to develop in order to collect the microbes at the scale and then process the microbes so that we can get at their small molecules. Now, one of the things we do that other people in the pharma world don't do is we are only discovery. We have found the box and the niche we do. We end up working with other companies that need these molecules, who will do the preclinical work, who will do the clinical work. So we don't have that large overhead of development of a molecule. We can work with people to bring these molecules to them. So what is it that we really do? Well, let's think about scale for a minute. If you were to mine something like lithium, would you use a coffee cup? That's the wrong scale. If you're trying to get at small molecules that are in nature at parts per billions and parts per trillions, you've got to start with more than a lab size quantity. And typically a lab is about 100 milliliters. We're talking about 200 million times that. That allows us to amplify these small molecules, get them into testing and screening. And our partners will be able to discover these new molecules. And then with these new molecules, they become the starting points. We can derivatize them. We can develop tremendous IP on those molecules. So we have proven technology here. We've found molecules for Lilly, IL-17, Leidos, CTLA-4, University of Rhode Island, a neuroinflammatory inhibitor. We have multiple peer-reviewed scientific papers on this technology. It is proven that we can do what we're talking about, and we end up getting it in one microbiome, nearly 100,000 unknown, untested small molecules. We're working with major organizations, including the entire ecosystem of Harvard, Stanford, Princeton. And then we're working with other companies themselves, Mercury Bio, and then national labs like Los Alamos National Lab. So what do we do and how do we make money? It is immediate revenue as we work with people on some of the molecules. And then as molecules advance, we'll get upfronts, milestones, and royalties. That's the large potential from what we do. The markets are definitely pharma, but they're including ag, sustainability, cosmetic, longevity. These small molecules in nature have worked throughout life sciences. And then the technology itself, ultimately, we're going to license. So what are we doing now? We recognize it's a tough market. We put 9 million of non-dilute, 12 million of dilute. But as you can see, the round we're raising is at a very discounted rate. We are ready to execute at scale and deliver these molecules. So we're raising 2.2 million. The terms, I think, are amazing. So if you're out there looking for something that's exceptional, we think we bring that to the table. Scale brings, like I mentioned, over 100,000 from one microbiome into discovery and research. Our major focus is getting the first licensing deals, including also moving forward on major grant opportunities with ARPA-H, DARPA, NIH, BARDA. We're working with partners that can help us write these grants. That's our major objective. That was a lot of information. I hope that there's some questions out there, and please reach out to me or to Chad. We'll set up an appointment and discuss where we're going with this. Hopefully there's a couple of pharma companies or ag companies on the line, and they have a pipeline issue and are thinking to themselves, maybe they can solve a pipeline issue. I can tell you we will, we can. Give us a call.